Hello, everyone, and welcome to today's seminar. It is my pleasure to introduce Kübra Narci to you, who will be giving the second part of the NGS Workflows series. And today she's going to talk about benchmarking. The first part was on standardization of NGS workflows. And if you have missed that webinar last month, then you're most welcome to go to our GHGA YouTube channel and watch it there. And part 2 will also be on the YouTube channel afterwards. So before I hand over the stage to Kübra, I would like to take the chance to announce the upcoming events at GHGA. So we will be later this year at the GPC conference, and Kübra and her team will be holding a workshop there. So if you're interested in workflows, please do have a look, register for the workshop, and say hello to her. The next upcoming webinar is on 1001 reasons for a data management plan, and that will be in collaboration with the Elixir Germany team, DENBI, and the seminar or the webinar will be held by Daniel Woodberg and Helena Schnitzer. And just a few words on GHGA. So we are the German Human Genome-Phenome Archive, and our mission is to provide GDPR-compliant data sharing of humanomics data. And this will then hopefully enable the secondary use of clinical omics data and also provide translation of these research insights into genomic medicine. And we can't do this alone. So diverse communities are involved in that, like computational biologists, geneticists, clinicians, physicians, and also patients who are all involved in helping us to set up this infrastructure. And if you would like to learn more about GHGA, then please do go to our website, ghga.de, and there you also find our training resources. And on YouTube, there is a list with all the webinars that we have held so far that you can rewatch if you've missed one. And if you like to stay updated with what's going on in GHGA, please do sign up for our newsletter. You'll find a barcode at the bottom of the page that leads you to the subscription page. Now I would like to introduce Kübra to you. Kübra is a bioinformatician at GHGA and part of the Workflows workstream for over a year now. Before that, she obtained her master and a PhD in bioinformatics from the Middle East Technical University in Ankara in Turkey, and has also worked as a bioinformatics analyst for 7 British Genomics. And today, Kübra is talking about benchmarking of NGS workflows. And with that, I hand over the stage to her.
Welcome, everyone, to the second part of our NGS harmonization webinar series. And in this second part, I will be talking about benchmarking of next generation sequencing workflows. And so our outline will follow that. First of all, I will quickly go over what GHGA goals and objectives are. Then in the first part, I will be mostly talking about what is benchmarking, its components, and why we need benchmarking. And after a quick questions and answers break, we will be talking about the importance of reachability of benchmarking results through the community. And then I will try to give the idea of continuous benchmarking and reflecting our standardized benchmark procedure in GHGA. And finally, I will show the whole process of benchmarking using SAREC variant caller as an example. The main goal of GHGA is to establish a federated national infrastructure and national platform for payer human omics data. GHGA is a complex and multi-stakeholder ecosystem which requires neutral and technically competent standards. These standards must be adaptable for disparate purposes and useful for the broad sense of end users like academia, public, clinical usage, and also commercial usage. Specific attention to policies of equity and diversity must be developed for all global communities. That is why the frameworks for data sharing, protection, and data analysis will be will be following ethical legal with fairness, which also means that we are GH4GH Global Alliance for Genomics and Health Compliance. Particularly as workflows teams in GHGA, we will be providing standardized procedures for analysis data and also for the workflows. In the previous webinar, I was mainly talking about harmonization in standardized GHGA workflows and how we will be providing portable, reproducible, scalable, and accurate analysis. Workflow management languages like Nextflow, SnakeMake, or CWL enables easily the process of constructing bioinformatic pipelines through good templates. And as GHGA, we are working closely with workflow communities in order to develop and also to maintain our FAIR pipelines. We are using package managers and software containerization tools in order to be able to run analysis in any infrastructure, ensuring portability. Our containers and pipelines will be shared through suitable registries like NF-Core or biocontainers. And in order to provide a full execution portability, workflow execution services and task execution services will be orchestrated through Kubernetes and also enabling data managers like EWS. Currently, we are hosting our pipelines, configurations, and tools in GHGA GitHub for stability and scalability. While workflow managers provide reproducibility for bioinformatic analysis, they do not guarantee accuracy, which is essential in biological data analysis. In order to ensure powerful and accurate analysis, it is a common practice to use state-of-the-art and best practice high-performance bioinformatic tools. Yet, it still needs to be investigated and proven to be an efficient workflow. A systematic assessment of these methods is called as benchmarking. And today, I will be talking about how we can be certain on the tools that we use and the principles of accurate bioinformatic analysis. And at the end of the webinar, you will understand the importance of accurate, efficient, and transparent benchmarks. And you will know the commonly used benchmark data sets, methods, metrics, and those especially in variant calling analysis. The aspects to consider for standard benchmark analysis will be more clear, and you will know the significance of community-developed, transparent, and objective platforms. In computational biology, resources are frequently faced with a choice between several computational methods for performing data analysis. While this represents an opportunity, it is a challenge itself, since method preference can significantly affect the conclusions by genomic context, since softwares, the methods may produce different results. Benchmarking studies aim to rigorously compare the performance of the tools using a well-characterized benchmark datasets to appoint the strengths and also weaknesses in the methods. So the scope of a benchmark should be clearly defined at the beginning of the study, since it fundamentally guidelines the design and also the implementation. In general, we can define three broad types of benchmark studies. First one is through the method developers. They usually perform tests in order to demonstrate the merits of their approach. Even they compare the performance with a couple of others. Usually it is not global and it is not enough to derive conclusions. More systematic comparisons are needed, and they are performed through neutral studies. either an independent group or collaborations with the method authors came together through the purpose of comparing a broad range of tools. Those studies can provide clear guidelines for users and highlight the actual weaknesses in the methods. And another approach is through community itself. In order to foster the comparisons, community challenges are being organized. Those generally concentrate on specific contexts like somatic variant calling, RNA sequencing, or single-cell sequencing in the purpose of finding the best performing tool for this specific analysis. Any group or method can attend to the challenge and be a part. Since the benchmark itself is not performed through the method authors, they are performed through the organization. The results itself are very transparent and objective, and the same for all the people. And at the end, they share the results through a dashboard for the end users, and everyone can check it. Benchmarking studies must be carefully designed in order to provide an accurate, unbiased, and informative results. We can break down the concept into 4, and I will be talking about them separately in detail, but in summary, benchmarking data refers to the data set which we'll be using for the test. Those data set serves a ground truth since their content is already known. Second component is the method to use for the evaluations, for the comparisons. Importantly, the analysis and the results format should be aligned with the others. It should be the same. Third component is performance metrics. At the end, we need a statistical measure in order to refer accuracy and precision to define how good the method is. Fourth component is about sharing the result, providing some charts and dashboards for making use of comparisons. Benchmarking starts with a set of well-known data. Its content is already clear, so that the software can be tested to see if the given the same input, if it produces the same output, or at least something close to that. And in especially variant benchmark analysis, variant calling studies, the data sets are the products of elaborately studied works, which include multiple sequencing techniques and multiple variant callers. Those data sets produced and approved to be a benchmarking reference data sets. And depending on the purpose, the data set can be either a real sample or a synthetic data or simulation. The produced data set can be called truth, baseline, or gold standard. They are usually accompanied with high confidence regions, which refers to the genomic region where the sequence is resolved well. And on the contrary, there are difficult regions, which are referring to the complex regions in the genome. And in genomic analysis, truth variance and high confidence regions are highly crucial to evaluate performance of both the methods and the sequencing technology itself. There are 2 kinds of benchmark sets. One is through sequencing real samples. Genome in a Bottle, GHGA dataset hosted by US National Institute of Standards and Technology is one of the commonly used one. In total, they have 7 samples. HG001 is a Utah sample. HG002 to 4 is an Ashkenazim trio. And HG005 to 7 is from Han Chinese family. The truth sets are prepared through 14 different sequencing technology with different library preparation methods, and well-characterized common calls between them are found using multiple variant caller algorithms available at that time. And the common calls are released as truth variant sets. Those samples are commonly used in the benchmark studies for variant calling analysis, since it is well known. Yet these consensus calls are usually in simpler regions, and they have tendency leading to overestimation of the true variant call performance. So that is a drawback. And the other major difficulty using these results, this truth calls, is that the fact that the variant calling algorithms evaluated against them were also used to construct the reference data set in the first place. So the results can be biased. Sometimes it is more helpful to use a synthetic or simulated data set. The synthetic deployed benchmark data set has the advantage of not relying on a consensus call set across the variant callers. For example, the synthetic deployed SynDip dataset is derived from de novo long-read assemblies of two homozygous human cell lines. It is uniquely advantaged to provide benchmarking data for more challenging regions, like duplicated sequences or tandem repeats. Simulated data sets or sequences also can be created programmatically using specific algorithms. Variations can be spiked into the regions by purpose, especially to the difficult ones. Yet we need to keep in mind that some of those variants in the sets might not represent the real genome very well, since it is simulated. Next component is about comparing the results. It is the method itself. Most importantly, neutrality should be maintained comparing the methods. And also we need to know that performance of the analysis could be impacted from the parameter settings and software versions. And most importantly, whatever we run, we need to record it through good provenance, keeping the history very well tracked. Sorry. Although it seems a non-trivial task, the major challenge in benchmark is to construct a method to see how good the software is. At the end, we need to see how our results are compatible with the truth set. And in variant calling benchmark analysis, the usual approach is to compare variants with the truth reference sets directly. That seems to be simple, especially for easy regions and single nucleotide variants through the genome. But basically, you need to compare all of the variants, including indels and complex variations itself. And for the complex regions in the genome, it is not that straightforward. Variant callers may represent variations in different forms, which we call them as ambiguity in allelic representation. Actually, normalization of the variant representation is required because of these complications. Here I want to show some examples. In the table A, a deletion of A in homopolymer region is shown in three different representations. It could be either positions. And in table B, multiple nucleotide polymorphism, a MNIP is shown. in either as 3 separate single nucleotide variation or one large substitution. And in table C, an insertion either can be one big one for base pair, or in 2 positions as 2 separate events. Actually, even the soft pairs referring to the same change in the genome, it can be found a mismatch just because of those differences in representation. And it could be reported as mismatch at the end. And some of those ambiguities can be resolved through specific normalization methods. But actually what we need, what is crucial, is to be aligned with the community and to have a standard method to compare those variants. Global Alliance for Genomics and Health, GH4GH, benchmarking team was working through the goal of providing some standardized benchmarking approaches to provide neutrality in comparing the soft pairs and tools. The team has worked towards solutions for several challenges. First of all, they were trying to define high confidence variance calls and stratification regions that can be used as benchmark. Second, they were trying to develop methods to compare variant calls, which will be robust to different representations. And also they are trying to standardize counting on the basis of matching stringency. And at the end, in order to produce a standard output, they were trying to find a format as VCF to evaluate the performance matrices. In the pipeline, they are using RTG developed comparison tool, VCF eval, which implements graph library. And variant normalization is done through VCF tools. And it also implements XCMap in order to help representing variance in a standardized way, like left shifting all the indels in repeat regions in order to be able to have those in the same position. And quantification is performed through Quantify and HappyPie tool from Illumina. And at this stage, the variants are labeled with annotation in VCF coherent format, which will be given as an input. The need of choosing the best tool for our analysis brings the need of measuring performance matrices. There are 2 sides of this story. First of all, you need to look from a technical side and think about qualitative matrices. And on the quantitative side, it will be important to know biological benchmarking these performance matrices for variant analysis. And before going details of quantitative metrics, I want to quickly mention on the other factors that may affect the software choice. Methods should be evaluated according to secondary measures as well, including runtime, scalability, and other computational requirements like memory and CPU usage. By experience, runtime and scalability may vary enormously between the methods. User friendliness, installation procedures, and documentation quality are also important, since no one would like to deal with hard-to-use softwares if the understanding process takes just too long. From the developer's perspective, code quality and use of software development best practices like unit testing and continuous integrations are very important, which prove the accuracy of the pipeline itself. Similarly, adherence to commonly used data formats like GTF, BET, BEM, SAM, or VCF files greatly improves accessibility and also extensibility of the tools. Yeah, before going into quantitative metrics, we can give the break.